Hey, Hammy here. This is video number 2 for chapter 3 over section 3.2, in which we'll talk about carbs or carbohydrates. First thing we want to look at is functions of carbohydrates. There are two main functions. When we think of carbs, we think of sugars. And what do you need sugars for? That's right, energy, right? Sugar makes you hyper, gives you lots of energy. One that kind of lesser known that a lot of people don't know about or think about as often is the structural role. In other words, using them as building materials in living organisms. Carbohydrates are CHOs. You know, CHO, down a road, CHO, not CHON, CHO. Okay, CHO is always 1 carbon, 1 hydrogen, 2 oxygen, 1. So the very common example, glucose, C6H12O6, it's a 1 to 2 to 1 ratio for most carbohydrates. And we split them in kind of the simplest to most complex: monosaccharide, disaccharide, polysaccharide. Anytime you see saccharide, that's referring to a sugar or a carbohydrate. Starting with the monosaccharides, mono meaning 1. So that is a single sugar, sometimes often called a simple sugar, usually 3 to 7 carbon atoms. Kind of the most well-known one that we talk about in biology is glucose or blood sugar. They are hexoses, hex meaning 6. So this is our C6H12O6. But we also, C6H12O6 can also be fructose or galactose. These are 3 of the common hexoses. Okay, they are, remember, what do we call it if they have the same structural formula but different, when you kind of draw them out, or same molecular formula but different structural formula, what do we call them? Isomers. If you said isomers, winner, winner, chicken dinner. Okay, they are isomers of each other. They have made up of the same amounts of atoms, but they're just rearranged and combined differently. The other common ones we'll talk about this year in AP Bio are the pentoses, which means 5 carbon atoms. So if you remember in DNA, RNA, ATP, we say there is a 5 carbon sugar, either ribose or deoxyribose. Those are monosaccharides, 5 carbon atoms, and very important molecules in living things. The next slide here, I just want you to be familiar with glucose, some of the other hexoses, how these are represented in organic chemistry, how you may see them illustrated in labs or scientific articles or textbooks. A here is sort of the true structural formula. So we listed all our elements: carbon, 1, 2, 3, 4, 5, 6 carbons, 12 hydrogens, and if you count 1, 2, 3, 4, 5, 6 oxygens. Okay, so that's kind of everything written out. Sometimes we simplify it a little bit. If you see a shape like this, at the corners here, at the corner here and here, that's going to represent a carbon atom. They don't write the carbon atom. It's assumed. If it's not, like here, the oxygen, they'll put an oxygen in there or a different element. Otherwise, all those corners are assumed to be carbon, 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 carbon. Okay. Then we can get even simpler, where we will just show the ring structure and we'll leave off all the H's and OH's. And actually, this little carbon, the CH2OH up here. Now understand that this right here is actually, this carbon has a hydrogen, and then an OH group. That's where that H2OH comes from. They're just condensing it so it takes up less space. So this is just a much simplified version. And then sometimes you'll see even simpler, where you'll just have this hexagon, you'll have the O in the corner, and we should kind of understand, or we'll realize, oh, that's probably glucose or galactose or fructose. Okay, next is disaccharides. You remember in the last video, we talked about taking a monomer and combining it with another monomer to get a polymer. Okay, in this case, we take two monosaccharides together. We're going to get a polymer called a disaccharide. It's di meaning two monosaccharides joined together by dehydration synthesis, and we'll look at that in a minute. Some common examples that you may have heard of: lactose. When you take glucose, simple sugar, galactose, that makes lactose, milk sugar. Some people don't have the enzymes to break that down. You hear them say, oh, they're lactose intolerant. Their bodies, and they get real sick on their stomach if they have too much ice cream or milk with their Oreos, and they just don't have the right enzymes to break that down. Sucrose is a common one. My Mountain Dew's got lots of sucrose or table sugar in it, which is glucose and fructose. Maltose is another one you may see that is 2 glucose. So glucose plus glucose equals maltose. Glucose plus fructose equals sucrose. Galactose plus glucose equals lactose. So 3 kind of common disaccharides that you'll see in biology. Okay, this slide is just illustrating how we do dehydration synthesis and degradation or hydrolysis. How do we combine monosaccharides together to make a disaccharide? And then how do we hydrolysis? How do we degrade or break that back down? So you see in the dehydration reaction that's going this way, okay, here's glucose plus glucose equals maltose. We take the OH off of this carbon atom. Remember, this is a carbon atom right here. And we take the H away from this oxygen. Okay, take those out. H, O, H, which is H2O. Take out a water. Dehydration. And we're going to use that bond there. Over here, you can see it. We're using that bond there to connect them together. So dehydration synthesis, syn, to put together. Okay. If we want to split them apart, there's other enzymes that will do hydrolysis, where we'll take an H2O, we'll take a water, put it back in, break those bonds apart between the 2 glucoses. Okay, and we'll go back this way. We'll add the OH group over here and the hydrogen to the oxygen, and we can go back that way. So just kind of an illustration. And for each of these biomolecules, we'll kind of look at an example of how that's done. Finally, our polysaccharides. They're made up of poly means many monosaccharides all strung together. And there's some examples that we use for energy. I'll put big E for energy. These other examples, I'll put S for structural purpose. Okay, the 2 energy ones in plants, the energy is starch. So as the plant is producing glucose and fructose through photosynthesis, and actually G3Ps, which we'll talk about later this year in the photosynthesis chapter, they'll string them together and store them as starch. Those are in fruits and seeds and that kind of stuff, so that when that plant starts growing, it has nutrients, it has energy, I should say, for that little seedling to start growing. In us, in animals, that's glycogen. Okay, we take extra sugars we consume. Our liver strings them together as glycogen. It's a little bit better short-term storage for an animal than just the plain glucoses. Structurally, cellulose, which is the most abundant organic molecule on Earth. Sometimes your book mentions that daily plants are making about 100 billion tons of cellulose on this planet daily. It's everywhere. We can't digest it. It's just fiber. Helps us make us more regular. Other animals that like rabbits and cows and other things that chew their cud, termites have special bacteria in their stomach that help break down that cellulose. For us, we can't break it down. It's just fiber. Chitin. Chitin is in fungi, fungal cell walls, and also exoskeletons of some animals. A lot of insects. When you step on that beetle and it goes, it crunches. Okay, that's the chitin. It actually has an antiviral and antibacterial type property. So we as humans have found ways to do that. We cannot digest it. If you eat a beetle, you can't digest their exoskeleton, their crunchy exoskeleton. Peptidoglycan is found in the cell walls of bacteria. Remember, that's how we tell the kingdoms and kingdom or domains of not eukarya, eubacteria, apart from the archaebacteria, whether they have peptidoglycan or not in their cell walls. And again, it's just a sugar. It's just a sugar. And notice peptido, okay, peptide often refers to a protein. Okay, that's because the monomers each have an amino acid chain attached to them. Amino acids we'll learn about are part of what make the subunit, the monomer of proteins. So those are attached to the sugar, so we get glyco, which means sugar, and peptido, which refers to the amino acid or the protein part. This slide just gives you a visual. Over here is starch. There's actually two types of starches. If it's unbranched, so see here, it's just all single, glucose is all wound together like this. Okay, that's called amylose. Okay, amylose. If they're branched, then we call it amylopectin. Amylopectin is more highly branched. About every sixth carbon, there's a branch of it, and it's more of a random pattern. Here, energy storage in animals, glycogen. Again, they're showing a picture of the liver where we store that. Look how highly branched that is. Okay, branches of little glucoses everywhere, and it's kind of stored in our liver, and we can release it into our blood as it's needed. Finally, they just wanted to show you a picture of more of the structural molecule, cellulose. And if you look at cellulose, you see these little microfibrils, these long chains of glucose. And it's a very good structural material because look what you notice here, between here and here. What do we represent with dotted lines? Remember when we talk about properties of water? Hydrogen bonds. There's actually some hydrogen bonds going on. So you got all these fibers of fibers of glucose molecules, and then they all kind of hold together. That makes a very strong structural molecule. Okay, so that kind of wraps it up for carbohydrates. In the next video, we'll take a look at lipids. This is Hammy, out.